May I request Professor Devinder Singh to start today's session?
Good morning to all the participants on this webinar on evolutionary biology. So this subject, evolutionary biology, is really a fascinating one that integrates various disciplines of biology that includes genetics, ecology, cytogenetics, paleontology, molecular biology. So all of them, they get integrated and we get this subject of evolutionary biology. Actually, it was Julian Huxley, who integrated all these branches into what he called as modern synthesis of evolution. And with growth, now we have got this full-fledged discipline of evolutionary biology, which includes studies in the field as well as studies in the laboratory. As far as laboratory studies are concerned, most of the work is being done on the wonderfly, Drosophila melanogaster. This fly has been responsible for research leading to 6 Nobel Prizes in such various fields as cancer, diabetes, autism, and in 2017 there was a Nobel Prize on the circadian rhythms. The first Nobel Prize on work related to Drosophila was given to the famous scientist Thomas Morgan, who gave the theory of inheritance, chromosomal theory of inheritance of characters. So this all makes the subject very fascinating for the students, for the researchers, as well as for the general public as well. And when we were thinking of organizing this webinar on evolutionary biology, which happens to be one of these subjects for our PG students, so we could not think of a better person than Professor Amitabh Joshi to deliver this webinar, who has worked a lot on this subject. He's rather a master of this subject as far as our country is concerned. So before we actually start his lecture, I'll request Dr. Bharti to introduce Professor Joshi to the audience. Thank you.
Thank you, sir. And it's my singular pleasure to introduce Professor Amitabh Joshi, an evolutionary biologist, population ecologist, geneticist, and a professor at Jawaharlal Nehru Center for Advanced Research, Bengaluru. He is an elected fellow of the Indian Academy of Sciences, National Academy of Sciences, then Indian National Science Academy. He is also a J.C. Bose National Fellow of the Department of Science and Technology. He has been awarded Shanti Swarup Bhatnagar Prize for Science and Technology. That is one of the highest science awards in 2009 for his immense contributions to biological sciences. Professor Amitabh Joshi is much concerned about the future of evolutionary biology in India and is instrumental in establishing the Indian Society of Evolutionary Biologists. Besides, he has a keen interest in literature, poetry, and is a great admirer of Urdu poet Allama Iqbal. Amitabh himself is a very good poet of Urdu, Persian, and English. Given this backdrop, it's always fascinating and captivating to listen to him as he elegantly and effortlessly creates, weaves, and articulates historical, philosophical, and poetic narratives into science. That is absolutely commendable. And at this point, this reminds me of a couplet. सोहन के सारे सलीके ज़बां में रखता है, सोहन के सारे सलीके ज़बां में रखता है, अजीब शख्स है नज़र में कलाम रखता है. So please join me in welcoming Professor Amitabh Joshi.
Thank you very much, Heminder ji, for the kind introduction. And it's a great pleasure to be here on this webinar supported by Punjabi University Patiala. I have very fond memories of my earlier visit there a few years ago when I had delivered a talk in person at the conference that Dr. Heminder had organized. So without further ado, let me start. So the title of today's talk is Evolutionary Biology in the Lab and Beyond. And I would like to sort of, cover two little different aspects in this talk, if I can have the next slide. Uh, this is the outline of the talk. So the first roughly half of the talk, I will try to give you some flavor of our work on experimental evolution. And my purpose here will be to try to illustrate the power of experimental evolution in the laboratory in order to address fundamental questions that arise in evolutionary biology. So today I will just focus on some of our work on one aspect of evolutionary biology in the lab. We work on several other things apart from what I will talk about today, and we also do experimental population ecology in the laboratory and so on. The roughly second half of the talk, I would like to switch gears and talk about the importance of evolutionary biology or an evolutionary perspective, not only to biology as an academic discipline, but also to society at large. Why evolutionary perspective or evolutionary biology is actually extremely important for us to solve many of the problems facing society, ranging from problems of environment and climate change to pandemics to other biomedical problems as well as agriculture and forensics and so on. And finally, I would like to end by giving you a little bit of flavor of some of the really fantastic work that has been done in India in the area of evolutionary biology. Unfortunately, this is often not very well recognized, but if one looks at biology as a whole, the most significant contributions from Indian scientists to the growth of biological knowledge have largely actually come in the area of evolutionary biology and evolutionary ecology. So I'd like to end by giving you a little bit of the Indian scenario and acquainting you with some of the wonderful work that has been done by different people in India. So as was mentioned, we use fruit fly Drosophila melanogaster as a model system to address questions in ecology and evolution. And since Professor Bharti mentioned my liking for Allama Iqbal's poetry, I cannot resist giving you one of my favorite couplets, which is also a very good rationale for experimental evolution and modern systems. So Allama Iqbal said in Persian, گفتند جهان ما آیا به تو می سازد گفتم که نمی سازد گفتند که برهم زد. In English, it is God asked me if I did find his universe to my liking and taste. When I said no, then he replied, create your own and lay this waste. So poets and mathematicians do have the luxury of creating their alternate universe to their liking and taste. For biologists, the best we can approximate that creation of a new universe is by using convenient model systems. So this is an attempt to create a well-defined, environmentally controlled universe of a single species and use that to address evolutionary questions. So I will first just briefly tell you how we maintain these populations. Adult phase is kept in these cages, as you can see in the center of the slide. And there is Drosophila food medium made of jaggery, bananas, and yeast, and ethanol, and various other things. The eggs would lay flies on the flies would lay eggs on the medium, which you can see in the bottom right hand side of the slide. And next slide, please. So what we would do is from these food media in the petri dish, we would cut out little strips of food with approximately 70 eggs plus minus a few on them, and then carefully place these foods into glass vials that have some food. So the larval stage is actually grown in these vials. Next slide, please. So within these vials, for each population, we may set up 40 or more vials, depending on what we are doing with that population. The larvae would feed and then grow up, and eventually they come out of the food, they evacuate their gut contents, and then they wander and often pupate on the walls. As you can see, some pupae forming and some wandering larvae on the vial to the right of the screen. Next slide, please. And finally, when the adult flies have emerged from the pupae, we collect those adults back into the cages and the cycle starts again. So all the experimental work that I will talk about today consists of two types of populations. There are the so-called FEJ populations, which are selected populations. There are 4 replicates of these populations. In these populations, as we will see in a little bit more detail, we are selecting for faster development and earlier reproduction relative to their ancestral controls. And the ancestral controls are called the JB. JB simply stands for Joshi baseline populations. These are populations normally maintained on a 21-day cycle. Next slide, please. So this selection for rapid development and early reproduction is done in the following manner. Under uncrowded conditions, which is what we are keeping them at, about 70 eggs per vial with 5-6 ml of food, Drosophila melanogaster takes about 200 plus hours to become adults from starting from egg at 25 degrees. So these 4 ancestral populations have been maintained in the lab for many hundred generations now on a 3-week discrete generation cycle. Discrete generation means that the next generation does not coexist along with the parents. So we collect the eggs laid by the parents, set up the next generation with those eggs, and then discard the parents. The eggs hatch into larvae and the next generation grows up independently of the parental generation. And from egg collection for one generation to the egg collection for the next generation, there is a 21 days or 3 weeks period. Now, in these control JB populations, there is no pressure on how fast they have to develop. All the individuals in principle can eclose to become adults and therefore contribute to the breeding pool of the population. There is no selection on the development time or how long it takes from the egg to the adulthood for these flies. And the egg to adult development takes about 9 to 10 days at this temperature of 25 degrees. Therefore, before we collect the eggs for the next generation, in the JB populations, there is about an 11 to 12 days worth of adult life phase that the flies undergo. And during this time, of course, the flies are reproducing, they are laying eggs in the medium, but those media are being discarded every alternate day. We are changing to fresh food. So those eggs are not becoming part of the next generation. Only the eggs laid on the day 21 from the previous egg laying date of the previous generation, only those eggs are becoming part of the next generation. So in our lab, way back in 1997, from these JB populations, from each of them, we established one new population. So we got from JB 1 to 4, we got FEJ 1 to 4 populations. And in these populations, once the pupae darken, we start monitoring the pupae very closely. And as soon as the eclosions begin in the vials, we collect only the first 15% or so approximately of the adults that eclose in each vial to become part of the breeding pool. So they are collected into the cages, and within 3 days of adult life, the eggs are collected from these cages to start the next generation. So in all other respects, the FEJ are handled identically to the JB controls, who are also their ancestors. Next slide, please. So this is just a graphic picture of the two different forms of maintenance in these two types of regimes. The JB controls on the top, you can see from day 0 to day 21. The eggs are in the vials, then they become larvae. Larvae pass through 3 larval instars, they become pupae, and eventually there is eclosion to adults. And then there is 11 or 12 days of adult life, partly in cages from day 12 to 18. On day 18, the flies are put into the cages from all the 40 vials of that population, and the food given to them is supplemented with yeast paste, which induces egg laying. And then the eggs are collected on day 21, and 40 new vials, each with 70 eggs or so, is set up to initiate the next generation. Adults are then discarded. So the adult phase, which is the red bar above the timeline for the JBs, that adult phase is almost 10-11 days long. By contrast, if you look at the selected populations or the FEJ, as you can see on the right hand side bottom of the screen, this is the distribution of development time, the distribution of eclosing flies, and only the first 20%, 15 to 20% of flies that eclose are actually collected into the cages to become the breeding population. So because we are taking only 15 to 20% of vials from flies from each vial, we set up much larger number of vials with eggs to begin with, so that the final breeding population size can be similar, around 1500 to 1800 flies in both the populations. This is done so that there should not be any difference in the level of random genetic drift that is undergone by FEJ populations compared to their control. So they must be similar in all aspects except the selection for rapid development, and secondarily the selection for early reproduction. Within about 3.5 days of becoming adults, we collect the eggs for the next generation. So FEJs are selected for two things: primarily to develop faster than the controls, and secondarily, having developed faster, to be able to successfully reproduce at adult age of 3.5 days compared to adult age of 10 or 11 days in the JB populations. And both of these selection pressures have some implications for the evolution of the system, which we will talk about. So this system, as I mentioned, was established in 1997 when I was a young assistant professor and the lab was just starting. Today, the FEJ populations are close to 800 generations of selection, and they are still going strong. Next slide, please. So the first set of results I will try to talk about today has to do with exemplifying something that often happens in evolution, which is that yesterday's adaptation becomes today's constraint as the environmental context or the selective context changes. So this is about the transient evolution of a non-optimal life history. Now, by life history, what we mean is the timing of onset of reproduction, the duration of reproduction, distribution of reproductive output, and eventual aging and death. So for the FEJs, if you think about it, given the selection pressure we are imposing on the lab that they have to develop fast and also be early reproducing, the optimal life history for them would be to develop really fast to adulthood. And one of the consequences of fast development is that during the larval stage, the flies or the individuals would have less time to put on weight and to store large amount of lipids, which would be required for supporting egg production or fecundity early in the adult life. So really fast developing flies are also expected to be smaller than the controls. But we also know that early in adult life, all else being equal, the larger females will lay larger number of eggs. So FEJs would need to be both fast developing, but also somehow try to be relatively fecund around the early adult life, a third day or so. So one could expect that an optimal life history for these populations would be to evolve perhaps a greater rate of weight gain during the larval stage, so that despite developing quickly and being relatively having less time to put on weight, they still manage to have a decent amount of fecundity. Now these four panels on this slide are showing you data from generation 30 of the FEJ selection. Left top slide is showing the adult lifespan or longevity, and you can see that FEJ longevity has reduced compared to their controls, which are the black bars, their ancestors. You can see that number of eggs per female on the right hand top panel in the FEJ populations has increased over 30 generations compared to the controls, even though their dry weight per fly has decreased, as you can see in the bottom left panel. Dry weight of FEJs has come down, yet fecundity has been increasing over the 30 generations of selection. And in the bottom right panel, you can see that indeed the eggs that FEJ females produce per unit dry weight is actually substantially greater than what is produced by their ancestors. So during the first 30 generations of the selection, these FEJ populations are indeed evolving or appear to be evolving towards their optimal life history. They have increased their fecundity in early life, even though their body weight has gone down, and that has been achieved partly by sacrificing the adult lifespan, which anyway for the FEJ populations surviving beyond 3 days of adult life does not improve their Darwinian fitness in any way, because after that new generation is initiated, adults are discarded. Then after the 30th generation, we were busy recording some other phenotypes, some larval behaviors and other things. And we returned to these traits, these life history traits, after 40 more generations had elapsed, which is in the next slide. Next slide, please. So this is for generation 70 data. And we were expecting that these flies would continue to evolve more in the same direction as we had seen 40 generations earlier. But to our great surprise, we found that during the generation between 30 and 70, life history of FEJs was seemed to be evolving in the opposite or non-optimal direction, which is an apparent paradox, because under natural selection, organisms should not evolve in non-optimal ways, such which would reduce their Darwinian fitness. What we see here is top left-hand panel longevity has come back up at par with the ancestral controls, even though, as we said, longevity does not affect their Darwinian fitness in any way as long as they survive for 3 days. Fecundity has reduced much more compared to the ancestors, which is not surprising, because if you see in the bottom left panel, dry weight has also continued to reduce, because development time has shortened a lot more between generation 30 and generation 70. These are now very small, very fast developing flies. But most interestingly, if you look at the bottom right hand panel, eggs per unit dry weight have reduced a lot compared to the controls. At generation 30, it was higher than the controls. So somehow, instead of sacrificing lifespan and producing more eggs per unit body weight to maintain some reasonable amount of fecundity despite reduction in body size, they seem to be going in the other direction, which doesn't make sense in terms of the theory of evolution. So we came up with a hypothesis, which I will discuss in the next slide, as to what might be going on. So one of the things that is known from a large number of studies on flies is that females can undergo a physiological switch between two different strategies, one of investing more of their resources, their lipid reserves, to high fecundity at the cost of lifespan when the lipid level in the fly's body is high. And on the other hand, when the lipid levels are low, which is the situation to the left of this graph, they actually invest more in somatic maintenance, resulting in higher lifespan at the cost of lowered fecundity. So this is the classic trade-off between reproductive output and somatic maintenance investment of lipid reserves, which is well known in Drosophila, and it is believed to be mediated by some as yet unknown mechanism by which flies are able to assess their lipid level. What is the mechanism of assessment? We don't know. Nobody knows. So we hypothesized that there is some threshold level of lipid in the body which acts as a physiological switch to trigger the shift from high fecundity, low lifespan lifestyle to high lifespan, low fecundity lifestyle. And we further hypothesized, as we show in the next slide, that somehow during the course of evolution, as we know, FEJs have become smaller. So on the right of the horizontal red bar, we assume there was some particular lipid content corresponding to the JB ancestors and FEJ at generation 0 when the selection began. And as FEJ began to evolve to become faster developing, they also became smaller. So their lipid content perhaps also decreased. But at generation 30, the lipid content was lower than the controls, but still greater than the threshold level that would switch the physiological state towards the high lifespan, low fecundity. We then hypothesized that sometime between generation 30 and generation 70, as the lipid level continued to decline along with the body size, it crossed that threshold level and the physiological switch got thrown. And now the FEJs have switched into a mode where they invest more of the lipid into high lifespan compared to fecundity, even though that doesn't benefit them evolutionarily. Basically, their yesterday's adaptation, which is believed to be an adaptation to fluctuating environmental conditions in the wild, has now become a constraint on attaining the optimal life history under the new selection pressure the FEJs experience in the lab. So this was our hypothesis. And the next slide, I show you some evidence that supports this hypothesis. So in these 4 panels, I will just tell you what they are. Maybe the x-axis may not be very visible. First of all, in each panel, there is data from eclosion day 3 and day 10. Please ignore the day 3, day 10 data. They are not relevant to today's story. We will focus just on the data at eclosion. So the top left-hand panel shows the absolute amount of lipid per flight, lipid content. And again, black bar are controls, white bars are the FEJ. As you can see, FEJ has much less lipid than their controls. The top right-hand panel shows the proportional lipid content as a percentage of total dry weight. And you can see that not only is the absolute lipid content of FEJs lower than controls, even the proportionate lipid content is less than controls. And we were later able to figure out the reason why this happens. This is because during the metamorphosis in the pupal stage, the FEJs actually burn lipid at a very high rate compared to the controls because they have to rapidly finish the metamorphosis, which is energetically a costly process, and lipid provides more calories per unit mass than the carbohydrate does. So up to the point at which the larvae become pupa, the percentage lipid content of FEJ and JB is actually the same. Total lipid is much less in FEJ because they are much smaller. But after that, in the pupal stage, lipid content falls even more. So it becomes difficult for them to preserve their meager amount of lipid at eclosion because of the primary requirement of developing really fast. The bottom 2 panels on the left hand side is the starvation resistance in hours. That is how long the flies can survive under starving but not desiccating conditions. And you can see that FEJs being smaller and having less lipid, they survive starvation for a shorter period of time than their controls. We know from previous work that starvation resistance is highly correlated with the lipid content, the total lipid content. The most telling slide or data in terms of our hypothesis is the bottom right hand panel, which shows the starvation resistance per unit lipid. Per unit lipid, FEJs have a starvation resistance more than 3 times greater than their controls. Significance of this is that it is again known from previous work on Drosophila that once the lipid is mobilized from the fat body to the ovary for the immediate future egg production, it cannot be returned back to storage under starving conditions. So once the lipid is mobilized into the ovary for
Sound. Hang हो गया इसको नहीं चल अपना
Is there any problem in?
hang.
Hi, I'm so sorry. I think my connection had dropped. So sorry.
Okay.
So to continue where we left off, once lipid is mobilized to the ovaries, it cannot be used to resist starvation. This last bottom panel on the right hand side therefore indicates that indeed the FEJs are retaining more of the lipid for somatic maintenance rather than committing it to reproduction, suggesting that this physiological switch, which is believed to have evolved in the wild in order to allow flies to adapt to fluctuating nutritional conditions, this has now become a constraint preventing the FEJs from attaining an optimal life history in the laboratory. So this is a very nice illustration of a general evolutionary principle. Next slide, please. Could I have the next slide, please?
Done already, Amitabh. Right.
So sorry, for some strange reason, I think my connection is not behaving today. The whole week I've been online in various things. It has so far been working fine. Okay. So the next issue I want to talk about with the same experimental system is that of sexual conflict and reproductive isolation. What we mean by sexual conflict is sexual conflict at the genetic or genomic level. And in particular, we are referring to what is often called interlocus genetic conflict because it involves different genetic loci in males and females. And this conflict arises because males and females have very different means of maximizing their Darwinian fitness through reproduction. Their reproductive strategies are quite different. The reason for this is that for males, the limiting factor for them to maximize the number of their offspring is access to females that they can mate with. Whereas for females, the limiting factor maximizing the production of offspring by them is not how many males they can mate with, but how many eggs they can produce. So it is actually access to resources, not access to males. This difference drives the evolution of traits in males by which males manipulate the female endocrine and reproductive systems via various small peptides, the so-called accessory gland proteins that are ejaculated along with the sperm in the seminal fluid. And these peptides are received by receptors in the female reproductive tract and they impinge on the neuroendocrine system in order to modulate the female's reproductive physiology in ways that is beneficial for that male which has mated with her, but may not be beneficial in terms of Darwinian fitness for the female partner in that mating. This then creates a selection pressure for females to become resistant to these manipulations by the males. For example, imagine if some mutant female arises in which the receptor does not bind these male sex peptides properly. Such a female will not be manipulated by the male and will have higher fitness than other females. So this sets up a coevolutionary arms race, which is the fundamental feature of this so-called interlocus sexual conflict at the genetic or genomic level. So here in the pictures, you can see we set up all four kinds of crosses between the FEJ males and JB females and vice versa in all combinations. So on the extreme left and the extreme right, you see the homotypic or parental type crosses, JB male, JB female. You can see males in Drosophila are about half the size of females. The middle two crosses indicate JB male and FEJ female. You can see the JB male is almost the same size or a little bit larger than FEJ female. And then you have crosses between very small FEJ male and a very large, relatively speaking, JB female. So the two bar graphs on the left show two types of mating choice assays. One on the left is mating assay in groups, which has a large number of flies of all four kinds, JB male, JB female, FEJ male, FEJ female. They are allowed to mate in a large chamber, and then we see how many matings of each type occurred. And the right-hand side panel is an individual mate choice assay in which one female is given a choice between either FEJ male or JB male, and you see which male, which type of female chooses to mate with. What you can see from the bars which depict the frequency of each of the four types of homotypic and heterotypic mating is that one type of mating occurs at very low frequency, and that is the mating between the small FEJ male and the large JB female. Basically, it is difficult for the FEJ male to successfully court and mount and mate with the much larger JB female. So that one type of mating actually happens at very, very, very low frequency. The other heterotypic mating, that is the large JB male with the small FEJ female, does happen at high frequency, but it has a consequence which is shown on the three panels stacked one above another on the right hand bottom side of your graph. So these panels depict on the x-axis time, hours from setup. The topmost panel are four setups where different type of females was given one male, and what you can see is that in one particular case the mortality or death rate of the female after mating increases greatly with time from mating. That is the topmost curve, and that again corresponds to the mating between large JB male and small FEJ female. And this mortality induced by mating with the large JB male shows a distinct male dose dependent effect if you go down those panels from 1 male to 5 males per female to 10 males per female. In the case of 10 males per female for this particular combination, almost 100% of the females are dead within about 1.5 to 2 days from their matings. So what we find here is evidence for incipient reproductive isolation between the FEJ population and the JB ancestors, because one of the two heterotypic mating, FEJ male and JB female, hardly occurs due to behavioral and morphological issues. The male is too small to successfully court and mate. The other kind of heterotypic or hybrid mating occurs at high frequency, but females die shortly after the mating. So effectively, no hybrid progeny or very few hybrid progeny are produced. So in fact, this was work done by a former student of mine, Shampa Ghosh, when she was doing a PhD. And we were able to show partial reproductive isolation. And we hypothesized that this may have something to do with the FEJs having evolved lower levels of sexual conflict. So we hypothesized that the FEJ females are no longer adapted to deal with the extremely toxic level of manipulative proteins produced in large quantities by the JB male in their seminal fluid. And another student of mine, Avani Mittal, who has finished her PhD recently and is now a postdoc in Sweden, she investigated this further. Next slide, please. So this is one experiment we did to preliminarily try to look at the relative effect of the genome versus the body size. So we created a third set of population called JC. This is created just for one generation. It's not a selection regime for this particular assay. And what we have done for JC is, by rearing the larvae at high density, we have made small flies that are otherwise JB in terms of their genomes, but their size is similar to the small FEJs. Okay, so JC is a small sized JB genome fly. And when we do the same kind of experiment where we give different males to the females of different types and record the mortality of how many females died over the first 14 days after the matings, on the x-axis are the female types. FEJ on the left, you can say that when FEJ female is mated with JB male, that is the dark, very tall bar, they undergo much higher mortality than when they mate with either the FEJ male or the JC male. This means that the JC male is not so toxic to the females compared to the JB male, despite having the same genome as JBs, only it has much smaller body size. So the male toxicity might be more a function of body size is the first indication. If we look at the JC females, the third set of bars along the x-axis, JC females are as unaffected by mating in terms of mortality induced as the JB females are. They are not behaving like the FEJs. So here it seems that for the female tolerance or resistance to mate manipulation by the male accessory gland proteins, perhaps the genome is much more important than the body size, because the dosage of accessory gland protein that comes in the seminal fluid might play a role in how toxic it becomes. So body size of the male is important, but whether the female can avoid binding those proteins may be more of a genomic effect. So this was the first indication, and we next decided to try to tease apart the effects of body size versus genome, and also try to address what aspect of the FEJ selection regime, whether the faster development and therefore indirectly the body size reduction is responsible for lowering the level of sexual conflict in FEJ population, or whether it is because they have very short adult life phase. Next slide, please. So if we again recapitulate the study system, there are two differences between FEJs and JBs. One is the FEJ are faster developing and therefore very small, body size is very small. Second is that they have very short duration of effective adult life. In other words, it is likely that the opportunities for multiple mating might be much less in the FEJ population, whereas they might be much greater in the JB, because there are 10 or 11 days of adult lifespan before the eggs are collected for the next generation. So females may be undergoing multiple mating. The implication of this is that the level of sexual selection through male-male competition for mating success may be much higher in the JBs compared to the FEJs, and sexual selection in the form of male-male competition is one of the things that is known to increase evolutionarily the level of interlocus sexual conflict. Next slide, please. Could I have the next slide? Yeah. So this slide just summarizes the breeding ecology of JB versus FEJ. Again, we're not going to the details of how this is measured. And these are arbitrary values on the y-axis. They have to be converted to actual matings. You can see that lifetime mating rate of JB is much higher than FEJ. Lifetime courtship rate of JB is much higher than FEJ. To cut a long story short, effectively what this means is average male in a JB population may be expected to mate about 5 to 6 times during its 10 or 11 days of adult life before the egg collection for the next generation. In the case of FEJs, the average mating success per male is about 1.1. So there are hardly any multiple matings. Therefore, it is true that sexual selection is also likely to be lower in the FEJ populations. So now, in order to tease apart the possible effects of reduced body size versus reduced sexual selection on the evolution of lower sexual conflict in the JBs, we set up two further selection regimes in which we relax different aspects of the FEJ selection. In the next slide. Next slide, please. So, in addition to the FEJ and JB, we set up two further regimes, RRF, which is relaxed selection only for rapid development, and RF, which is a complete relaxation of FEJ selection. In other words, it is going back towards the JB regime. So in the RRFs, what is happening is, instead of only choosing the fastest 20% who become adults, all the flies that eclose are collected to become the breeding pool for the next generation. But the second part of the selection, getting only 3 days for adult lifespan, that part is not relaxed. So RRFs are selected for only early development, but not rapid development, only early reproduction, but not rapid development. RFs have got relaxation of selection both for development time and adult life. They have a long adult life, so presumably they can have more sexual selection also. RRFs are no longer under selection for body size through development time, but they may be expected to still have a relaxation of the sexual selection.
नहीं नहीं yes, yes. Phone.
Sorry. So if RRFs show different evolutionary pattern with regard to sexual conflict traits, that would allow us to tease apart the relative role of body size evolution in the FEJs versus sexual selection in affecting the lower level of sexual conflict. Next slide, please. Can you hear me?
Yes.
Okay. I was getting some strange flicker on this.
No, it's okay. Oh, it's okay.
Okay, so RFs and RRFs have a huge difference in the adult phase of the lifespan, and therefore in so sorry, this is becoming embarrassing. Next slide, please. So this is just to show that in terms of development time on the left-hand side panels and adult dry weight on the right-hand side panels, the reverse selection has had an effect. These are from two different generations, I think generation 30 and 60 or something on the top and bottom. So development time of RFs and RRFs has increased relative to FEJs. They are going back towards JPs, and their body size has also increased. Most importantly, the rate at which the response is happening for these traits in RFs and RRFs is not different from each other. Next slide, please. So now we look at directly something relevant to sexual conflict, which is the size of the testes and accessory glands relative to thorax length, which is a surrogate of body size in Drosophila. And this is because we know that sperm competitive ability is determined by sperm length, which is correlated with testis size. And we know that monogamy adapted males, where there's very low sexual selection due to only one mating, they often have reduction in testis size, and the accessory gland size is related to how much accessory gland proteins the sex peptides are actually produced. Next slide, please. So if we compare mating duration and how long from eclosion to first mating in the RFs and RRFs, again, RFs and RRFs are deviating from FEJ from which they have been derived, going towards JB values, but more importantly, the difference between RF and RRF in either mating duration, which is believed to be correlated with how much accessory gland proteins are transmitted in the seminal fluid, and in how long they take to attain maturity, which could again be an indication of differential sexual selection. There is no significant difference between RFs and RRFs, at least for the first 50-60 generations of relaxed selection. This again suggests that perhaps sexual selection differences are not playing such a major role as the body size is in determining the JB-FEJ difference. Next slide, please. This shows the, on the left panel, the accessory gland size, the absolute size of accessory glands. On the right hand side, accessory gland size as a fraction of body size, which is the square of the thorax length. That is just to make the units of area equal. And again, the important thing to focus on right now for today's talk is that there is no difference between the RFs and the RRFs. So overall, what this suggests is that contrary to what we might have thought, sexual selection differences between JB and FEJ don't seem to be playing a major role in the reduction of the sexual conflict in FEJs. It seems to be more driven by the reduction of body size in the FEJs as a result of the faster development. And I hope that this sort of exemplifies with experimental evolution how nicely one is able to really go underlying details of what is the cause for the evolutionary changes that we are seeing over many, many hundred of generations of selection in the laboratory. Next slide, please. Okay. How much more time do I have? I suspect I've been going slowly.
No, it's okay. It's okay, Amitabh.
Okay. So now the second part I wanted to talk about, which is evolution is central to biology. This I'll just sort of illustrate this by a few quotes. In particular, I'll draw your attention to the last quote because that is not by an evolutionary biologist who could be accused of bias. Dobzhansky was an evolutionary biologist. Peter Medawar was actually a Nobel Prize winning immunologist and medical doctor who got a Nobel Prize for his seminal discoveries regarding the major histocompatibility complex in mammals and humans. And he says something very strong. He says, for a biologist, the alternative to thinking in evolutionary terms is not to think at all. This is a very strong statement. And indeed, what this statement exemplifies is that evolution is not a branch of biology, the way anatomy is a branch of biology, or molecular biology is a branch of biology, or physiology is a branch of biology, or biochemistry is a branch of biology. Evolution is a perspective or a logical framework within which we can examine all facts in biology, whether at the organismal level or the suborganismal or cellular or molecular level. It is the overall logical framework by which we make sense of the diversity of organisms, of processes, of forms that we see in the living world. So in some sense, biology without evolution would be like chemistry without the periodic table. It would just be a collection of diverse facts with no underlying logic to why that diversity of facts is the way it is. Next slide, please. In fact, one could even make a case that there are two kinds of biologies. See, we are just used to thinking of biology as one subject for historical reasons, whereas we think of the non-life sciences, physics and chemistry, as two different subjects, even though there is a lot of overlap and fuzzy boundary between them. Many areas of chemistry and physics totally interlap and mix and also inform each other. Similarly, there are actually two very distinct kinds of biologies that are asking different kinds of questions. One kind is asking the question, how is it that living organisms are actually alive? How are they taking energy from the environment, carrying out various processes of life and so on? And in this kind of biology, the questions are typically posed at the suborganismal level, at a molecular or cellular level. And they are mechanistic questions about the processes by which living things are characteristically living. But in addition to that kind of question in biology, we also need to explain why living forms are so diverse. Why are all living forms not just one kind? Why is that diversity occurring in patterns of clear relatedness, which is the reason why we can actually have a classification at all? And why life forms or species seem to be well adapted to the lifestyle and ecological context within which they live? This second set of questions is really addressed in what we might call broadly evolutionary biology, and these questions are typically posed and addressed at the level of an individual organism functioning in an ecological context, which includes both the abiotic environment as well as the biotic environment, conspecifics as well as heterospecifics. And in some sense, these two kinds of biology are actually very different from each other in the kinds of questions they ask. In terms of tools, of course, evolutionary biology uses all kinds of tools ranging from mathematical modeling to molecular biology to biochemistry to behavior to quantitative genetics to population genetics to you name it, biochemistry, anatomy, morphology. Everything is deployed to address the question. But the difference from molecular biology is that the questions in evolution are focused at the level of an organism functioning in some environmental context. Next slide, please. So that was regarding the centrality of biology, of evolution to biology as an academic discipline. Now I'll just briefly like to talk a little bit about the centrality to understanding and solving many human and societal problems. Again, on the slide, you might not perhaps be able to necessarily read the titles of these books. This is just to indicate the range of application. The book on the left top is Why We Get Sick, the New Science of Darwinian Medicine. And again, I would just say that whether it is infectious diseases or the so-called lifestyle diseases, evolutionary biology has a lot to do with how we choose to tackle them. The next book from the left is DNA Technology in Forensic Science. All of you are familiar with, for example, DNA fingerprinting. Now, in order to generate a DNA fingerprint from DNA collected from a crime scene, of course, you need molecular biology techniques. But in order to interpret the results you get and to assign a probability of how likely it is that your suspect is indeed the same person whose DNA was found at the crime scene, you need a lot of molecular population genetics or molecular evolution too. The third book is called The Moral Animal, Why We Are the Way We Are, the New Science of Evolutionary Psychology. And I would request all the students, if you have any interest, please just Google things like evolutionary medicine or Darwinian medicine or evolutionary psychology. You would be amazed at how much work there is in those fields. The fourth book is Darwin's Spectre, Evolutionary Biology in the Modern World, a very accessible introduction both to evolutionary biology in its basic concepts and its applications to, for example, medicine, agriculture, as well as human cognitive abilities. The fifth book is actually by two of my mentors, Larry Muller and Michael Rose. It's called Does Aging Stop? And this is about the profound changes that have happened since 1990 in our understanding of aging and therefore how to deal with aging as a result of the application of evolutionary biology to understanding aging. The last book on the right is an example of applications to agriculture, genetic mapping and marker assisted selection. The graphic on the bottom left of your screen shows what we now understand of early modern human migrations out of Africa. This has happened because of our ability to extract DNA from very old skeletons and look at genetic markers in 10,000, 20,000 year old skeletons. So this way we are able to trace the movement of humans before the recorded history or civilization began. And this sheds a lot of light, among other things, also on how the Indian subcontinent was peopled during the last maybe 40 plus thousand years. And on the right hand side is the kind of output you get from genome-wide association studies of looking for correlations between various genetic markers and various complex phenotypes, complex disease phenotypes in human biomedical genomics today. And again, the interpretation of these results, the results are generated by molecular biology techniques, but the interpretation of these results requires a non-trivial application of evolutionary theory. Next slide, please. This just lists many of the problems you are all familiar with. Today we are suffering from a pandemic of COVID, coronavirus. New pandemics, how they arise, why a pathogen switches a host from some other animal to humans, how pandemics spread. The whole epidemiology of new pandemics is essentially application of evolutionary biology and population ecology. The evolution of multi-drug antibiotic resistance in bacteria is basically an evolutionary problem. These are not problems that are going to be solved by molecular level interventions. In order to understand what is going on, we also need a very strong evolutionary perspective, aging, complex diseases, understanding human history migrations, epidemiological consequences of global climate change, a very, very important area of applied ecology and evolution today. How to optimize and manage phenotype by environment interactions in crop breeding, and many social problems. For example, why is domestic violence asymmetric by gender? Actually has very sound evolutionary explanation. Does monogamy benefit males and females equally? Has very interesting evolutionary explanation. And in many of these social aspects, the reason this is important is that the explanation provided by an evolutionary perspective tends to be very different from the classic sociological explanations or psychological explanations for many of these social pathologies. And therefore, if the evolutionary explanation is correct, and for some of these issues, the preliminary data suggests that that is likely, then it suggests that society may want to rethink how we try to deal with and manage some of these social problems. Because the sociology, psychology paradigms are beginning to look like perhaps they are not very correct compared to the evolutionary biology insight into the root cause of many of these social problems. So all in all, I just wanted to say with this is that often it is thought that okay, maybe evolution is important to biology, but it's of academic interest only. It has no applications. On the contrary, it has profoundly important applications for how we deal with many societal problems and also how we understand ourselves and the genetic makeup of our society, the historical development of our society, how we look at ourselves as a people in the context of the subcontinent and the world is also very greatly affected by an evolutionary perspective. Next slide, please. So I just now like to come towards the conclusion and talk a little bit about evolutionary biology in the Indian context. Evolution is very poorly taught in our school and university curricula by and large. There might be a few exceptions. And much of the material is actually wrong or out of date by decades. The state of the typical average university curriculum in evolution in India today is as if you had a physics curriculum in which there was no mention of Einstein, no mention of Max Planck's work, no mention of quantum mechanics, basically no mention of physics after 1925 or 30. That is the scenario with evolutionary biology, and it's really a shame. There is also very little in the curriculum to suggest to students that evolutionary biology is a very active and very creative research field internationally today. It's not by any means a dead field, and it also has great applications. The last point I would wish to make is that today, especially in India, we give an imbalanced emphasis to molecular biology. We equate modern biology with molecular biology, but ultimately Molecular biology is no doubt interesting, important, and powerful. Evolutionary biology is equally, if not more so. And to relegate essentially one half of biology to complete neglect is asking for trouble in terms of our ability to deal with societal and biomedical and agricultural problems, environmental. Next slide, please. This is particularly ironic because in terms of fundamental contributions to biological knowledge, our small community in India of evolutionary biologists has done far more than most of the rest of Indian biology has actually done. In fact, if you really think about it, after the Ramchandran plot, there has not been any major fundamental contribution to biological knowledge from India's molecular biologists. And Professor Ramchandran was not really even a molecular biologist, he was basically a physical chemist. Uh, the second point is that evolutionary research is not very expensive. And in principle, even state university, college level faculty could do world class research in evolution if they had a good idea of what is the state of the field and what are the interesting open questions in the field. Instead, across India, what we have is in small colleges, small universities, which may not be so well funded as research institutes, lot of biology faculty are essentially doing second-rate molecular biology work. They are hampered by lack of equipment because they are not aware of what are the range of questions available in evolutionary biology, which they could address. In many cases, we could benefit from the fact that we have many biodiversity hotspots. If one wanted to do field-oriented evolutionary biology and ecology research, India is paradise to do it in. And again, for that, we need faculty and therefore students who are the ones who grow up to become faculty to understand what is the state of art of ecology and evolution research and what are the open questions so that we can compete on a level playing field. When we do evolutionary biology and compete with our counterparts in more developed Western countries, in richer countries, we are not hampered by lack of money or lack of equipment. It's our brain against their brain. It's a fair competition. If we are doing molecular biology in India, which is a very technology intensive field, we are always competing against people with better access to, cutting edge equipment and much more money than we have. It's a very unfair competition. And therefore, it's a tragedy that instead of playing to our strengths in biological research in India, we are choosing to play to our weaknesses and overemphasizing molecular biology as a research field and in our teaching and in our curriculum. Next slide, please. So I'll now briefly just introduce you to some interesting evolutionary biology work from India. This is Professor Raghavendra Gadakkar from Center for Ecological Sciences, Indian Institute of Science. He is one of the world's leading experts on the social evolution of primitively used social hymenoptera. And one of his major works was to extend the work of William Hamilton on the evolution of altruism by emphasizing that it's not just relatedness which is important to determining social evolution of new sociality, but also ecological and demographic factors. On the left is a very well-known book of his, Social Biology of Rupalidia marginata. He has also written a more popular book on basic evolutionary thinking aimed at students called Survival Strategies, which is widely available in India. And I would certainly recommend it to all the students who might be listening. Next slide, please. This is Professor Uma Shankar and Professor K.N. Ganeshaya, who built their careers in the University of Agricultural Sciences in Bangalore. Uma Shankar was in the Department of Crop Physiology, Ganeshaya was in the Department of Plant Genetics and Breeding, and on the side, they did absolutely world-class work in evolutionary ecology. They were the pioneers of extending the concept of parent-offspring conflict in an evolutionary sense to plants. The concept was originally developed in animals. They were among the pioneers who extended it to plants. They have also over the years done fantastic work on one of the canonical textbook cases of mutualistic coevolution between pigs and pig wasps. And indeed, their work substantially changed our understanding of this paradigm example of coevolution. It is internationally recognized, extremely creative and wonderful work. Very, very well recognized internationally for their pioneering work in this field. Next slide, please. This is Professor Milind Vatve, who used to be at Abasaheb Darware College, Pune, and was later for several years at the ISER Pune. He's a microbiologist, but also works in evolutionary biology and ecology, a very versatile scientist. He is one of the very, very few people in India working on evolutionary medicine and came up with an evolutionary interpretation of type 2 diabetes, which leads to a somewhat better integration of unexplained aspects of this disease and also some very new suggestions for behavioral rather than medicinal interventions. And some of these behavioral interventions are now actually in controlled clinical trials at the BILD, which is established and functioning for last few years in the Dinanath Mangeshkar Hospital and Research Center in Pune. And a few years ago, he published this very interesting book called Doves, Diplomats and Diabetes, a Darwinian interpretation of type 2 diabetes and related disorders. One of the few examples of evolutionary medicine work being pursued in India. Next slide, please. This is Professor N.G. Prasad, who is in your neighborhood in Mohali, ISA Mohali. He uses Drosophila to address questions in sexual conflict and sexual selection theory, and is among his lab is among the top 5 labs working in this area worldwide. They have, among other things, taken the first comprehensive experimental validation in the same experimental system of all the various interlinked predictions from theory of sexual selection and genomic level sexual conflict. And last year, they actually provided the first experimental evidence of something that had been theoretically predicted, that interlocus sexual conflict, which we discussed earlier, could be a potent driver of reproductive isolation. Next slide, please. This is Professor Sutirth Day, who is at the Indian Institute of Science Education and Research, Pune. He does experimental evolution with both fruit flies and bacteria. He and his students did some of the first experimental evolution studies of evolution in fluctuating environments, environments which are not constant in the lab, but which are fluctuating along multiple environmental axes like pH, salt concentration, and oxidative stress. And they showed that bacteria that evolved under fluctuating stresses evolved very different adaptations compared to those that evolved under a constant stress, one stress at a time. And more interestingly, they actually showed that populations adapted to fluctuating environments also evolved resistance to antibiotics which they had not been exposed to at all. As a byproduct of adapting to multiple stresses that were fluctuating randomly, these E. coli populations acquired multi-drug resistance. And this is very interesting because it provides potential explanations of why people are finding that bacteria in nature, isolated from pristine habitats like high altitude mountain streams where there's no human habitation, bacteria isolated from deep inside the Amazon rainforest, from areas populated by no modern day humans, no civilized humans, only extremely, extremely, tribes that are still following natural. These are places where there is no way that the bacteria could have been exposed to second, third, fourth generation antibiotics, but they show resistance to all of them. And this work, again, the first of its kind in the world, opens up the possibility that it might be a byproduct of just evolving in response to fluctuating environment. Next slide, please. Finally, I'd like to say something about something that 4 of us do. 2 of them you just met, Professor Prasad and Professor Sutirth Day, and myself and my colleague at JNC, Professor TNC Vidya, who works on elephant social organization in the wild. We get together from time to time in a group called Foundations of Genetics and Evolution Group, and we work on fundamental issues in conceptualizing evolutionary biology and genetics from time to time. I would just like to draw your attention to one paper we published in 2017, Gupta et al. In the Journal of Genetics. This paper is ranked by Altmetrics in the top 5% of most discussed papers across all disciplines, science and non-science, on the internet across the time that they have been monitoring. It is also the first non-Western contribution to fundamental conceptual issues in evolutionary theory till date. Evolutionary theory, fundamental conceptual issues and debates have been dominated by people from Europe, from North Africa, North America, and Australia. And this is a group that is informal, friendly. We enjoy working together. We all have our own lines of research. We don't take any funding from anybody. This is just science done for the love of it and for the fun of it. And again, we are able to contribute non-trivially to the growth of evolutionary understanding in the world. Next slide, please. This is something I cannot resist talking about because I myself do experimental evolution. Experimental evolution in India actually began in the 1980s with the work of Professor Ranganath in Mysore University Zoology Department. And they did amazingly good work. He has now retired some years back. Modern experimental evolution began in the 1970s with the work of Larry Muller and Michael Rose, who you can see in this picture. Both of them were among my mentors when I was a PhD student and postdoc. And here you see both of them in this picture with Professor Prasad and Professor Day. This is not a very well-known fact, but you know, in India, we often say, where are the world leaders in biology? And the answer is we look in the wrong place. You won't find world leaders in biology, in molecular biology, you'll find them in evolutionary biology in India. If you look at laboratories in the world, research groups in the world doing long-term experimental evolution studies of the kind I talked about earlier in the talk, more than half of them are in India. This is a level of domination in a subdiscipline that you will not find for any other area of biology at all. And I list here, these are in the University of Mysore, my own institute, Jawaharlal Nehru Center, in Delhi University, in IISER Pune, in NCPS Bangalore, IISER Mohali, IISER Berhampur, Kalinga Institute of Technology, Bhubaneswar, and Ashoka University. Next slide, please. So I'd just like to end with saying that the need of the hour in our country is the inclusion of proper, detailed, and up-to-date training in the concepts and principles of evolutionary biology at both school and college level, and proper training in evolutionary biology basics for all biologists, regardless of specialization, at the postgraduate level. This is very important. You might have read articles in newspapers recently. Many leading doctors and medical people in India wrote that we have very few good epidemiologists in India to deal with problems like the current pandemic. Now, why do we have very few good epidemiologists? Because the foundational basic research underlying epidemiology are sub areas of evolutionary biology and population dynamics, population ecology. If you don't teach the fundamentals, the applications are not going to suddenly materialize. You know, good practitioners of the applications will not materialize. So evolutionary biology is important not only to train new evolutionary biologists, but also to train biologists in diverse fields to better make them able to deal with biological problems in a more holistic and sensible manner. And it's really important, in my opinion, that we must have at least one full-fledged national research institute for postgraduate training and research in all major areas, basic and applied, of evolutionary biology or evolutionary sciences more broadly. Next slide, please. I will basically conclude with that. Thank you very much for listening so patiently and putting up with the unnecessary dropping of the connection so many times, which caused a bit of delay. Thank you very much. And thanks to the Department of Zoology at Punjab University for inviting me to give this
talk. Thank you, Professor Joshi, for your thought-provoking talk, particularly last 4 or 5 slides were very interesting, and they must be inciting our students to leap in this field of evolutionary biology, because there is, as you have proved, there is a lot of scope for research, and not much resources are required for this type of research. And I think our PG students, they will take you from this talk, and those who want to go for research after their PG, they should think about this area of research as a potential area for PhD or for the PDF or whatever they want to.
Yeah.
That Joshi, quite a good number of students, they are going to ask some questions related
by all
to this.
means. By all means.
I'll start one by one. First question is that when you talk about FEJ and JB,
yeah,
when males and females, for example, when FEJ females mate with JB males, the females die.
Yeah.
And when FEJ males and JB females try copulation, the FEJ males cannot compete.
Yeah.
So it's a very apparent type of reproductive isolation. You have called it as partial reproductive isolation, but it seems to be quite better reproductive isolation that we see even among some good recognized species where hybridization is taking place. So why is it considered only as partial when it's almost sure?
The reason for considering it partial is that although even in a competitive situation or a choice situation, proportion of matings obtained by FEJ males with JB females is very low compared to all the other 3 types of matings. It is maybe 3-4% rather than 25%. Okay. But there is still 3-4%. So small number of matings do occur. It's not that totally none of the male is able to get a mating. Secondly, females die over a period of time. When FEJ female mate with the much larger, much more toxic JB males, they don't die immediately. Some of them survive for 10 hours, 12 hours, 24 hours also, 36 hours. So they do have opportunity to lay few eggs, and those eggs are fertile. So there is some amount of reproduction, much less than the other homotypic mating, but some amount of reproduction is still possible. It's not a complete.
Did you try to see that effect of reproductive in the F2, F3, F4 generations? When
yes, up to F2 there is no effect. F3 we have not checked. Up to F2 they are fine. The F2 are also easily formed. They are all fertile and so on. And in fact, for most of the body traits and life history traits, the F1 is roughly intermediate between the 2 parents. So it looks like a more of an additive scenario.
Regarding this JC, which they were very small sized individuals, of course, it's a survivorship strategy among the flies to produce small sized individuals when the food is in short supply or time is less for completing the development. So is there any crucial size which must be attained to survive among the larvae?
Yes, absolutely. So all holometabolous insects have a certain minimum critical size, which is usually attained somewhat early in the final instar of the larva. And unless they attain that minimum size, they cannot successfully pupate and complete the development. But in most cases, if food is abundant or otherwise situation is fine, they continue to feed long after attaining the minimum size and finally pupate at a much larger size. In the FEJs, because of the selection on rapid development, what has happened is the duration of third instar is much vastly reduced. And essentially, they attain the minimum critical size and then they stop feeding and they go for pupation. This we have also followed up in terms of the neuropeptide F signaling and so on, because this transition from feeding to wandering stage is accompanied by a reduction in neuropeptide F levels in the brain. And in FEJs, the neuropeptide F levels are very low even in the early third instar and second instar compared to their ancestors. Now, what happens in the FEJs is also the minimum critical size. I didn't show those data here. The minimum critical size itself has evolved to become smaller also compared to their ancestors. So it's a double thing. The effective critical size is less, and also immediately after that, they are just hardwired to go for pupation. And with the JCs, what we tried to do was to mimic exactly the size of the FEJs.
As you have mentioned that our knowledge about evolutionary biology can provide some solutions to even our social problems, like you have mentioned, is rape just for sex, or it is for dominance, or other social problems, because humans are now more guided by cultural evolution rather than biological evolution, as we see during this time. How you correlate evolutionary biology, knowledge about evolutionary biology, with the current situation, the problem that the world is facing, the COVID? Is there anything evolutionary biologists can suggest about from our solution about COVID?
Well, see, when we talk about solutions are of many types in terms of time scales of the problem. In the immediate term, given that we now have a new virus which has shifted to humans from some animal species and has now spread all around the world. See, one of the basic issues with understanding these things, so that we may not be able to do anything to turn back the clock with regard to COVID. For COVID, all we can now hope for is either a drug or a vaccine, and of course certain changes in behavior to ameliorate the person-to-person transmission. But in the longer term, see, COVID is not the first virus which has switched from animal species to humans, and it's not going to be the last. There we need to address two aspects evolutionarily. One is, what are the ecological factors that tend to promote a pathogen, virus or any other pathogen, bacterial, other pathogen, to switch hosts? Why does that happen? How does that happen? And in particular, what are the ecological factors that enhance the probability of switching to human hosts? And many of those factors are connected with modern day society. One is, of course, the increasing encroachment of humans into earlier pristine habitats and the destruction of habitats. There is a very close link between that and this host switching. The other is due to climate change, uh, the distribution range of many species, both hosts and potential hosts and pathogens, is changing on a year-to-year time scale, on a fairly fast time scale ecologically. So, for example, that was the case a year ago with the Nipah virus outbreak in Kerala. It was partly driven by the environmental degradation and the change in the, the host distribution of certain kinds of bats. So, also a lot of these kinds of markets where different species are housed together in close proximity to each other, alive, under very sort of overcrowded conditions, that greatly facilitates the movement of virus between species. So all these things, the evolution of host range expansion is a classic evolutionary biology topic that would be taught if evolutionary biology was properly taught in our schools, colleges and universities. Similarly, transmission, or the evolution of greater or lesser virulence. These are classic topics within host pathogen coevolution. And so do we expect a new pathogen that is spreading to become more virulent or less virulent? Depends a lot on the mechanisms of transmission from one individual host to another and various other factors. So there is no clear prediction that invariably a new disease will evolve to become less virulent. That's not always the case. They can evolve to become more virulent also. So all these aspects in some sense require a very integrated view of organisms, whether the pathogen, because let us not forget pathogen is also an organism which also evolves. Hosts are also an organism and there's an ecological dimension and ecology, when human is the host, then ecology includes everything we do as a society also. That is also part of our ecology. So one needs to take a very holistic view of these things. And this kind of training is really lacking in India to a great extent.
Harman Randhawa wants to ask a question that what type of traits should be selected to study reaction norms and phenotypic plasticity? What type of trait should be selected?
Well, one obvious intrinsic answer to that question is the trait would have to be plastic to begin with. Preferably plastic along multiple environmental axes that perhaps you can control in the laboratory. So it would be interesting to study reaction norms not just against one environmental factor at a time, but multiple environmental factors in a factorial design. And by doing that, one can also understand a little bit about how a reaction norm vis-a-vis one environmental factor, let's say reaction norm against temperature, is itself affected and its shape may change depending on some other factor like moisture level or crowding or something else. Beyond that, I think it depends on what traits you are interested in looking at, because there are so many traits which are plastic. There is also a question as to whether one can select for increased or decreased plasticity. Whether plasticity level itself is an evolutionary trait which can change over evolutionary time. And people have done this kind of work long back with, quite a long back with Arabidopsis and few other systems. There is work by this guy called Carl Schlichting from the 1990s onwards. They actually tried to see if they could select for increased or decreased level of plasticity in response to some particular. So there's quite a lot of work on plasticity as an evolutionary factor that goes on both with plant systems and with animal systems and also with microbial systems.
First year student Harsimran, she has got a very inquisitive question that whether a favorable trait in a population in some individuals will definitely lead to natural selection and evolution, what we call as evolution.
Well, when you say favorable trait, if you mean that it is a trait which helps the individuals carrying it to better survive and reproduce in their given ecological context, then the minute you have a variation which says that some individuals' traits are more favorable than the traits of the other individuals, then by definition natural selection is occurring. Now the question is, if natural selection is occurring, that is, some individuals with certain traits have higher Darwinian fitness, better survival, better reproductive ability than others, will it lead to adaptive evolution or not? There is no guarantee there. You can have selection, but for example, if these variant traits among the different individuals, if these variations were not heritable, if they were not transmitted to offspring, and by heritable, I don't necessarily mean that they are under genetic control. They can be culturally transmitted through learning also. They are still heritable. They can be ecologically transmitted through shared environment between parents and offspring. That is also a form of being heritable. But if the variations are not heritable at all, if the offspring don't resemble the parents any more than they resemble a randomly drawn individual from the population, then although selection is acting, evolutionary change will not happen in response to it. So for natural selection to occur, all that is required is that there be variation in Darwinian fitness among individuals in survival and reproduction. But for it to lead to evolutionary change in the next generation, in the subsequent generation, those variations in Darwinian fitness must also be heritable, whether by genic or non-genic means, doesn't matter.
Number of students they have asked in one form or
Sorry,
another.
I'm not getting your audio clearly.
Some problem with the audio?
I'm not able to hear you clearly. I'm sorry.
Hello?
Huh,
Is it
that's
okay?
better. Yeah. Now I'm getting.
So number of students they have asked one question in some form or other, but I combine it together that our knowledge about evolutionary biology, can it help in developing a vaccine against the novel coronavirus?
It does have some connection. Of course, the development of a vaccine against any virus lies completely in the domain of immunology. It's a very well defined, very focused branch of biology. But see, a vaccine that is developed against one strain of a virus is developed against that strain in that area at that particular point in time. But the virus is also evolving. So, vaccine development takes on the nature of a co-evolutionary arms race. If you consider the vaccine as an extended phenotype of the humans, which is a valid way of treating it, then it is a co-evolutionary arms race because vaccine is there. So there is now going to be selection pressure on the virus to change its or to have mutations in its surface proteins that the vaccine is targeting. And if the mutation arises with a new kind of surface protein which this vaccine cannot target, then that mutant virus will spread fast, and after some time that will become the dominant variety, and your vaccine will become useless against it. So you will have to then tweak a new vaccine. So it is a very much a co-evolutionary process that goes on.
Is there any relationship with the number of eggs laid with the size of the female?
Yes, absolutely. All else being equal, larger females tend to lay larger number of eggs. And it is a relationship that actually rises quite steeply as size increases. Eventually, it tends to level off, but the rise is quite steep. So the difference in 24-hour, how many eggs are laid within Drosophila fed to the same degree, but females with about a 50% difference in size, number of eggs can range from 2 or 3 in 24 hours to about 80 in 24 hours.
That is probably all from the student side, but I may add here for Dr. Joshi that in our PG classes, we have got a compulsory course on molecular biology itself, and very few universities in the country, they have that course. So,
nice. No, in fact, I am aware of that because I have visited your university also, and Professor Hivendra Bharti is a very good friend of mine. We often discuss, issues of teaching and how to promote evolution and so on. He is also one of the very active members of our Evolutionary Biology Society in India.
Okay, now over to Dr. Bharti for the final words and the vote of thanks.
Thank you, Amitabh. That was quite intoxicating, and it is always fascinating to listen to you. And it was quite, it was wonderful for the students as well, because I have been discussing with them whenever I have been teaching evolutionary biology, that if you want to learn something or listen to somebody, Caligula in evolutionary biology, it would be Amitabh Joshi. So at the end, I cannot resist quoting Allama Iqbal again. जहां में अहले ईमा सूर तो खुर्शीद जीते हैं, इधर डूबे उधर निकले, उधर डूबे इधर निकले. Thank you.
क्या बात है, क्या बात है, बहुत बहुत
Thank you very much.
थैंक यू very much.